So in this short video we're going to talk about hemostasis, or vascular response to the vessel injury. So when vessel gets damaged, those are obviously endothelial cells, and that's basement membrane. When vessel gets damaged, and I'm going to kind of represent what happens here, that some kind of a vascular damage here. Several major molecular mechanisms are involved in the hemostasis. First of all, injured endothelial cells start to produce thromboxane A. This is the molecule that leads to the first step in hemostasis, which is vasoconstriction. Vasoconstriction limits the blood flow to the damaged area and reduces the blood loss. Now, platelets can be activated by thromboxane A or adenosine diphosphate, that both molecules are released from the damaged endothelium, and they also can attach via surface receptors to the molecules that are now exposed at the injured endothelium, which are von Willebrand factor, preselectin, collagen. The main point that I want to make for you here is that platelets get activated, bind to the injured endothelium. Binding of platelets also leads to platelet activation. So you have two mechanisms. One is activation of platelets in the circulation, another activation through binding to the surface molecules. These activated platelets activate, so that's activated platelet. They activate other platelets. These activated platelets activate more platelets. So it's obviously a positive feedback loop. That activated platelet can activate other platelets. Now it looks like an avalanche of a response, but the key point, of course, here is why platelets stick only to the damaged endothelium and do not bind here. Well, it turns out that healthy endothelial cells, they release other molecules such as nitric oxide or various prostacyclins, which prevent platelet binding to the healthy endothelium. They work as sort of anti-coagulative molecules. So vasoconstriction, second, what we just described, the circular activation of platelets and formation of the platelet plug. And the third step of hemostasis, third step of the vascular response to injury, is coagulation. And we're going to talk about coagulation shortly. So let's talk about coagulation for a bit. So there are two pathways. There is an intrinsic pathway and extrinsic pathway. So what are the key differences between them? Intrinsic is activated by the damaged endothelium. So basically the damage is sort of contained inside of the vessel. So damaged endothelium. While extrinsic pathway is activated by the tissue. So the trauma is significant enough that it permeates the blood vessel and leads to the tissue damage. So that's what activates it, tissue damage. So intrinsic pathway has more steps. And since it has more steps, it is slower than extrinsic pathway. So what's important to understand, both pathways, intrinsic and extrinsic, they converge on so-called common pathway. So let's, in our next little fragment, we're going to chat about intrinsic pathway, then extrinsic pathway, then common pathway. First, we're going to talk about the intrinsic pathway. So in the intrinsic pathway, the endothelial injury activates first coagulation factor, which is factor 12. The asterisk means that it's activated. Activated factor 12, in turn, activates factor 11. Factor 11, together with a couple of other factors like factor 7 and calcium, activates factor 9. So at this point, it's important to understand. So there are a couple more things. We're going to put them in the brackets, that you need factors, activated factor 7, and you need calcium here. Okay? But factor 9 activates another factor, which is factor 8. Now you have these two key molecules, this one and this one. Factor 9 and factor 8 together form a complex. Let's put it this right here. Okay? So this complex between the 9 and the 8 is essential for the initiation of the common cascade. So, extrinsic pathway. In this case, you have some kind of a tissue injury, not endothelial, but tissue injury. Injury to the tissues that are outside of the vessel. Tissue injury leads to the activation of the generation of the tissue factor, release of tissue factor, and tissue factor itself activates factor 7. So factor 7, as you remember, is essential for the activation of factor 9 in the intrinsic pathway, and it also forms a complex with the tissue factor. And this complex here, this complex right here, is crucial for the activation initiation of the common cascade, which will be a topic of our next. So let's talk about the common pathway. So common pathway is activated either by the complex of activated factor 9 plus factor 8, that's from intrinsic pathway, or the complex between tissue factor and activated factor 7, that's from extrinsic pathway. So what they do, either of them can activate factor 10. Okay? An activated factor 10 activates 5, and 10, 5 calcium and phosphatidylserine form a molecule called prothrombin activator. Before we move any further, I know that there are a lot of words, a lot of numbers, so what you need to know specifically for the upcoming exam. Sequence 12, 11, 9, 8, 9 and 8 form a complex, that's for intrinsic pathway, and tissue factor activates factor 7 forming a complex, and then activate common pathway as well. So these two basic sequences would be more than sufficient. Again, 12, 11, 9, 8 for intrinsic pathway, tissue factor, and factor 7 for extrinsic pathway. And then common pathway. So let's talk about common pathway a little bit more. So common pathway, we start with prothrombin activator. Prothrombin activator. Obviously from the name you can guess that prothrombin activator activates prothrombin into thrombin. And thrombin, sometimes called coagulation factor 2, activates fibrinogen that, when activated, forms fibrin. And this fibrin here, those are essentially threads that enforce the clot formed by platelets in the first place. They hold platelets all together.